Hey everybody, so this is the second video that we are working on your data from the Enzyme lab. So we did the time series, we're looking at that. One last thing before we move on from that, let's talk a little bit about what other elements should be here. I think I mentioned briefly before talking about how you need to be careful, like when this if you're putting this into a paper that's in black and white, now we're submitting these on Blackboard, so it should keep your original format, but you need to think about, like, will these actually look like different lines? So the blue and red might not. So what we can do is we can click on one of our lines and we can, it's only doing one, of course. But what we can do is we can go in here and the fill will fill the data points, but then if you hit outline, it will do the actual lines themselves. And I don't know why it's not. There we go. Now it's selected all of them. So if you do outline, you can change it to a different one now. Changes everything else for you. So this one here, even if this was now printed in black and white, you would be able to see the difference between that blue and that black line. Okay, just one of those things to think about when you're doing this sort of thing. The other thing, if we go back to our example data, is we got rid of the title up here. What you're going to have is a figure caption. So figure 1, line graph of the amount of p-nitrophenol produced per minute. So in our graph, we would have blue is pH 5, black is pH 7, over a 15-minute reaction between silabiase and glucopyranoside. Right? A nice descriptive figure caption, and it tells you everything that's going on in this graph. Okay, so we would want that for this if you're going to use that in your paper. So one more thing that we want to look at. Get this to move around. Are excuse me, these averages. So we want to look at p-nitrophenol, our two averages for pH 5 and pH 7. So we're going to go and we're going to insert again. So this time we're going to do our bar graph again. Let's change up some of the, let's see. Let's change up our colors here. Let's go with something in a nice taupe. And let's do something that'll still see. There we go. So I changed that so it's you can still see the difference in the colors. It's just that you can actually read your designations in there. Right? So that's something. If this is too dark, you might not be able to read that. So you got to think about that. So remember, we're going to get rid of the chart title and the grid lines. All right. And so just like before, you would add your axes titles. So again, it would be nanomoles of nitrophenol per minute for your y-axis, and then this time, and it would be pH for your x-axis. Right? So don't forget to put those in. But the thing I wanted to work on was showing you how to do the error bars. All right. So if you just go over this, it adds in error bars. The thing is, Excel adds in error bars that are for the entire data set, including both averages. So this is just now the standard error for the entire overall data set. Now, I have no idea when you would actually use that, or why you would want to have that in there, but it's not the right one. It's not the one we want to put in there. So we're going to add error bars, but we're going to go in and we're going to go to more options. Okay, and we're going to go. The other way you can do that is if you do, I think it's design, yeah. So you can do add chart element, error bars, and then select more error bar options. Now, if you had what you had in there before, where it doesn't show this, you can select this. It looks like a little bar graph. So it might show you these other ones. So if it's showing you that one or this one, you can click on here and it'll give you the options you want. So we're going to go with error bars that go in both directions. It has a cap on it. All right, so that's all fine. What we want to do is we want to go in here and hit custom, and then we're going to specify the value. Okay, so our positive error and our negative error are going to be the same. So we just come over here to standard deviation and we click and drag to highlight that standard deviation. And then we come over here to click and drag the same standard deviation for the negative error value. We hit OK, and it should show our standard deviations. Now, this, besides the fact that our data doesn't conform with what we expected to happen, the fact that these standard deviations are huge. Our standard deviations are larger than, when you see this, the standard deviations are larger than the actual data set. That's telling you that there's probably something not right there. Okay, so that is how you add the custom standard deviations in. Don't forget to do your axes. In fact, we've got time. Why don't we just go through and do that? So you're going to go here. All this stuff usually can be in, let's see, is it format or design? Yeah, all this stuff can be accessed through the chart element as well. So if you want to add access titles, all right, we can add the primary horizontal, we can add the primary vertical, or we can do it through here, right, adding our access titles there. This is small n, big M for nanomoles of P nitrophenol per minute. And then this one, we'll call it substrate pH. Okay. So that now has all the elements you need for your graph. So what I'm going to give you guys is I'm going to post this data, this same data sheet. It's going to have basically everything except that you are going to have to generate your own averages, your own t-test, and do your own graphs. Okay. So this data is what you're going to be using. What happened there? Why does that look like that? That's not right. Okay, that's just a weird zooming thing. I don't know what's going on there. So I'm going to ignore it. Anyway, I think because I'm selected up here, it did weird things to it. All right, stuff like that happens. Generally, delete it, start over. That'd be found to be the best way to do things. So yes, so I will post this on Blackboard. You will have all your data here. You're going to have to generate your own means and standard deviations and your own graphs, as well as do the t-test yourself, so that you have that for the paper. So this is going to be what you are going to be writing your results section about and your discussion about. All right, we'll talk more about that. Okay. Hopefully this clears everything up. If you guys have any questions, don't hesitate to let me know. Again, I'm sorry we didn't get a chance to go over this in class. I felt like it wasn't really being effective, so hopefully this works for what you guys need and will help you figure out how to do things. If not, let me know, and I'll work with you until we get things figured out. All right, hope everybody's doing well. And